Good day to all of you. This video is about the notes on ELISA. So ELISA, the enzyme linked in immunosorbent assay, is a very sensitive immunochemical technique which is used to access the presence of quantification of a specific protein, which may be the antigen or antibody in the given sample. So it is carried out to detect and measure antibodies, hormones, peptides, and proteins in the blood. The principle of ELISA is based on the immunochemical principle of antigen-antibody reaction, and the several enzymes have been employed for ELISA, including alkaline phosphatase, horseradish peroxide, and beta-galactosidase. So it has three principles, that is, first is the immune reaction, then enzymatic chemical reaction, and signal detection and quantification. So sample may be the serum, plasma cell, etc. Enzymes are included like alkaline phosphate, horseradish peroxide, and beta-galactosidase. Next is the types of ELISA. So basically there are four types of ELISA based on the binding structure between the antigen and the antibody. So first is the sandwich ELISA. It is used to detect the antigen. So here is the figure in which firstly the microtiter well is taken, which is coated with the antibody. Then it is washed and adding antigen to be measured, sample antigen. Again it is washed and then adding enzyme conjugated secondary antibody. Again it is washed and then adding substrate and color is measured. And this color is directly proportional to the antigen in the serum. So in this way we can detect the amount of antigen with the help of its color. It has advantages too, that is flexible and sensitive. Antigen doesn't require purification prior to the measurement, and it is high specificity. Second type is the direct ELISA, and it is used to detect the antigen. So in this, the sample antigen is coated well is taken out, and then it is washed, and then the adding of enzyme conjugated antibody to be measured, and then it is washed, and then adding the substrate and color is measured. So it is somehow similar to the first one, that is sandwich ELISA, but it is less specific than the sandwich ELISA. So third one is the indirect ELISA, and it is used to detect the antibody. So process here are here, and in this process the antigen coated well is taken out, and then it is washed, and then then the sample antibody is taken to be measured, and then adding the enzyme conjugated secondary antibody. Then it is again washed and adding substrate and measuring the color. The fourth one and the last one type of the ELISA is the competitive ELISA, and it is also used for the antigen detection. So it helps to detect the antigen concentration in a sample, and more the antigen in the sample, more the primary antibody will bind to the sample antigen, and there will be a smaller amount of primary antibody available to bind to the antigen coated on well. So here is the process. So in this, firstly the incubation of the antibody with sample antigen is taken out, and then the antigen antibody mixture to the antigen coated well, and then it is washed, and then adding the enzyme conjugated secondary antibody. Again it is washed, and then adding the substrate and color is measured. So character of the competitive ELISA is the commonly used for the small molecules, even when the specific and detecting antibody is present in relatively small amounts. So application of ELISA is widely used for the quantitative examination and the qualitative detection. So quantitative examination of hormones, amino acid, growth factors, tumor markers, and qualitative detection of the bacterial or viral antigen antibody against microbes. So advantage of the ELISA is to detect antigen or antibody present in very small quantities in tissues or blood. It is highly economical and a sensitive method. It is non-isotopic immunoassay, that is, an enzyme is used as a label, and there is no risk of radiation hazards, more sensitive than RIA. So it is about the ELISA and its types. Thank you.